Hello and good afternoon, everyone. We'll give folks a few more minutes to roll in here and we will get started. Thanks everyone for joining. We'll give it about one more minute and then we will get started. Okay, it is 1:02. I want to be respectful of everyone's time. Thank you all for joining today. Just want to confirm that you all can see my slides, correct?
Looks good.
Wonderful. Thank you. All right. So, like I said, hello and welcome to the Invasive Mussel Collaborative webinar series. We have some excellent speakers who have agreed to share their work with us today. Presenters Kennedy Zwarych, who is an alumni with the McGill University, will be sharing their work regarding the limited coexistence of zebra mussels and native mussels in the Richelieu River over the past 25 years. And Sasha Kataev with SUNY Buffalo State will be presenting their work on multiple invasions decimate the most imperiled freshwater invertebrates. My name is Nicole Angel, and I'm joined by my colleagues Sam Tank and Autumn McGowan with the Great Lakes Commission. And on behalf of the IMC, we just want to thank you for participating in today's webinar. So before I turn the presentation over to our presenters, I just wanted to provide some background on the collaborative. So the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of invasive Dreissenid mussels in order to produce measurable and ecological and economic benefits. The Invasive Mussel Collaborative published a management strategy in 2018 to reduce invasive mussels and their negative impacts. So to make progress on these goals and this strategy, the collaborative established 4 different work groups, which you can see here on the screen. The research work group, the Dreissenid toxicity testing work group, the coastal site priorities work group, and the most recently established planning and implementation work group. So next, I'm just going to briefly highlight each of these work groups just to give you a sense of what kind of work the IMC does. So starting with the research work group, this group was charged with coordinating existing research and developing targeted plans and projects that address Great Lakes management priorities. The work group recently released a mapping tool called the Dreissena Project Coordination Mapper, and it allows researchers and managers to share their work and collaborate with others for the advancement and protection of waterways from invasive mussels. And this work group is soliciting additional research and applied control projects, which can be submitted directly through the mapper. Moving on to the toxicity testing work group, this group was charged with reviewing the strengths and weaknesses of mussel toxicity control methods used in various labs to identify and make recommendations to further refine and standardize these methods. So the studies reviewed for this work have been compiled into a table that provides a searchable collection of literature for Dreissenid laboratory toxicity and open water control testing methods and is available on the IMC's website. Additionally, a manuscript detailing the recommendations made can be accessed using that bit.ly URL on the screen here. Excuse me. Okay, moving on to the coastal site priorities work group. This group was charged with reviewing information on where Dreissenids are impacting Great Lakes resources, evaluating sites based on this information, and then developing and implementing a system for prioritizing sites for potential management activities. The work group released an interactive dashboard and map that displays nearshore data from the Coastal Site Priorities Index. The tool is called the Great Lakes Experimental Mussel Site or Suppression Site Screening Tool. It's a tongue twister, but we call it GLEMST for short. And this features a dashboard and associated indices, which are intended to inform future applied Dreissenid research and management activities by aiding in site prioritization. And then finally, moving on to the planning and implementation work group. This work group was charged with developing a research and management roadmap for the site and regional scale mussel control tools. They're also working to identify stakeholder and funding priorities and remaining knowledge gaps to guide future Dreissenid control activities. And this work group is really building on the products and outcomes of the other 3 work groups. So you can be on the lookout for more exciting work from this work group over the next coming year. And then just some background on the IMC and their communication network. So this webinar is part of the communication network, and the purpose of them is to facilitate learning and information sharing on topics of common interest. The webinar will be recorded and archived along with past webinars on the IMC's website. And if you want to learn more about the collaborative or interested in staying in touch with our communication networks, we do encourage you to visit our website and join our listservs. And then finally, while we always strive to minimize technological issues during these webinars, we do apologize for any that might arise. Before we get to our presentations, I just want to cover a few logistical items. So we've automatically muted all attendees to reduce interruptions, and attendees will remain muted with their webcams disabled throughout the webinar. But we do accept and encourage written questions from attendees during the webinars. So if you have any questions, please use the Q&A function to do that. Questions can be submitted anytime during the webinar and will be addressed after all the presentations have been given. And then additionally, if you're experiencing any technical difficulties, please use the chat box for that, or you can email my colleague Sam. Her email is my spiel. It's now my great pleasure to introduce our first speaker, Kennedy Zwarych. She completed her master's in Dr. Anthony Riccardi's research group at McGill University in Canada. Her thesis focused on long-term changes of unionid communities after Dreissenid mussel invasions, specifically in the St. Lawrence and Richelieu Rivers in Quebec. She is broadly interested in conservation biology and freshwater, marine, and terrestrial systems, but is especially passionate about researching invasion biology. Kennedy is currently publishing the results from her thesis while working as an environmental scientist at Nova Scotia Power. And with that, I will stop sharing my screen, and Kennedy, you can take over screen sharing, and I will pass it along to you. We are very excited to hear your presentation. Thank you.
Thank you very much. Bear with me while I pull up my presentation.
Absolutely. I'll stay on and I'll give you confirmation that it is working properly.
I appreciate that. Okay.
There we go. It's looking great.
Perfect. All right.
Go off camera. Thank you again.
No worries. Okay, now I need to make sure that I can see it. All right. So thank you everybody for tuning in today and spending your lunchtime here. Or if you're in Canada, thanks so much for taking the time out of your holiday to be here. Today, I'm going to be talking about one of the most conspicuous impacts of the zebra mussel, and that is their impact on native freshwater mussels. Freshwater mussels from the family Unionidae, which I'll refer to as unionids for the rest of the presentation, have very different life strategies than those of zebra mussels. They can live up to 100 years in some areas of North America and have a typical lifespan of about 20 to 30 years in the area of Quebec in Eastern Canada, where I conducted my research. As filter feeders, they filter large amounts of nutrients in the water column and eventually deposit them into the sediment. Since unionids burrow into the sediment, they continuously keep the sediment aerated and mixed, and effectively create islands of hard substrate in otherwise soft and sandy areas at the bottom of lakes and rivers. By doing so, this creates habitat for other benthic species. There are many, many different species of unionids in North America, However, they are also one of the most endangered faunal groups. And this is partly due to the introduction of the zebra mussel. Zebra mussels attach themselves to hard substrate, including the shells of unionids, as you can see here, which we call fouling. So specifically with unionids, fouling impairs the unionids' ability to burrow into the sediment, to feed, respire, and get rid of waste, since oftentimes zebra mussels are so dense that they prevent the unionid from opening and closing their shell, as you can see in the fouled mussel on the right-hand side of the photos. So unionids keep themselves upright in the sediment in order to siphon nutrients from the water column, but sometimes the weight of attached zebra mussels can be so great that the unionid can't keep itself upright anymore and will either fall over or deplete its energy resources trying to maintain stability. Some studies have shown that given choices of substrate, zebra mussels prefer to settle on live unionids rather than other inanimate substrate, and that the body condition of those zebra mussels, so things like shell growth, body mass, and glycogen content, or their energy stores, are enhanced when they are attached to unionids rather than inanimate substrate. Zebra mussels require dissolved calcium for growth, osmoregulation, and their reproduction. Zebra mussel populations tend to be largest in North America when calcium concentrations are above 25 milligrams per liter in North American systems. This is likely because of their evolutionary history. As a Ponto-Caspian species from Eastern Europe, they evolved in very ion-rich waters. So naturally, it makes sense that over here, they also do well in high calcium concentrations and ion-rich waters. In waterways with high calcium concentrations, those that support large populations of zebra mussels, unionids often suffer substantial mortality or near total extirpation within about 4 to 8 years following zebra mussel invasion, shown here in the red. Unionids, though, can sometimes exploit habitat refugia to escape excessive fouling from zebra mussels and ultimately coexist. With regards to calcium, zebra mussels require concentrations that far exceed those required by North American unionids. North American unionids tend to only require about 2 to 3 milligrams per liter, and with zebra mussels, this is much higher. So that being said, there is the possibility for the coexistence of unionids and zebra mussels in areas with certain particular water chemistry. So between about 15 and 20 milligrams per liter, the population abundance of zebra mussels and their impacts can really vary substantially. Some risk assessments consider water bodies below 20 milligrams per liter to be at low risk for zebra mussel invasion. However, there is a lack of research investigating the impacts of zebra mussels over long periods of time, especially over decades, in these suboptimal conditions. A great place to study this research is the Richelieu River in Quebec, Canada. This river has a consistent calcium concentration of about 18 milligrams per liter for its entire length, which greatly exceeds the amount that local unionid species require, which is, like I said, about 2 to 3 milligrams per liter. The Richelieu River was invaded by zebra mussels in 1996 after its headwaters, Lake Champlain, became infested around 1993. The colonization of the Richelieu is assumed to have been driven by a constant supply of larvae exported from Lake Champlain. We used quadrat sampling by SCUBA surveys to estimate unionid densities and fouling metrics at 7 different locations throughout the river sporadically between 1998 and 2023. So here's what we found. Unionid densities declined exponentially over time from approximately 17 per square meter in 1998 to less than 1 individual per square meter in 2023. This data is similar to patterns commonly observed in high calcium areas, although the main difference is that this occurred over a 25-year period instead of the typical 4 to 8 years where you see huge drops in unionid populations and very small populations after that. So we fitted this data with a linear mixed effect exponential decay function, which is shown in the black line, and we used random effects lines by site, shown as the semi-transparent colored lines. We also observed the number of unionid species to decrease from 10 species to only 4 over this period of time. To put that in context, remember that these mussels have a lifespan of about 20 to 30 years, and they don't start reproducing or they don't reach reproductive maturity until they're about 10. So over 2 generations of unionids, they've been reduced to less than half of their original diversity. We measured fouling intensity using 2 metrics. The first one simply being the number of attached zebra mussels to each unionid, and the second being the mass ratio of attached zebra mussels to the unionid. That one takes into account the size and weight of each unionid and zebra mussel. While using both of these metrics, we found that fouling levels remained relatively low over time compared to other calcium-rich waters that were invaded. All of this being said, why did we see such intense high mortalities of unionids when fouling levels weren't very high? So it's possible that this could be a situation of multiple stressors. So we had some thoughts and explored some alternative hypotheses. The first one is climate change. Die-offs of bivalves elsewhere have been attributed to extreme temperatures caused by climate warming. But to our knowledge, no major die-offs of unionids have been associated with summer heat waves in the Richelieu River. Also, the critical thermal max for unionids and their host fish were higher than the average and maximum surface water temperatures measured in the Richelieu between the past 20 years-ish, making it unlikely that the mortalities that we saw in unionid populations were caused by this. Unionids require host fishes in order to reproduce. Their larvae, called glaucidia, have a developmental stage where they attach to the gills and fins of fish until they are old enough to, until they become juvenile mussels and drop off. Declines in fish populations could possibly contribute to unionid mortalities, but to our knowledge, there have been no major die-offs of fish within the Richelieu River in recent decades as well. It's also important to note here that many species of unionids found within the river are fish host generalists, meaning that many species of fish would need to be completely extirpated from the river in order for unionid populations to be affected by this. Another potential cause of long-term unionid decline could be the installation of dams, which can alter water flow rates and alter sedimentation. So there are 2 dams that exist on the Richelieu River, although both of them were built in the 1960s. So given their age, they are unlikely to have caused the steep declines in unionid populations that we noticed in the early 2000s. Many areas of the Richelieu are also subject to industrial and agricultural pollution. However, testing for PCBs and DDT showed levels, low levels throughout the river. So it's difficult to definitively claim that it really affected unionid populations in the early to mid 2000s when we saw those steep drops, but wasn't significantly impacting them before or after this time. So lastly, other biological invasions could be contributing to the loss of unionids. For example, tench and water chestnuts are both Ponto-Caspian species from Eastern Europe and invaded the Richelieu River in the early 1990s, along with the zebra mussel. These are unlikely culprits of causing mass unionid mortality, since neither of these species have been reported to harm unionids in their other invaded ranges within North America. So with all this being said, a potential explanation for the declining populations of unionids that we saw in the Richelieu River is food competition in combination with sustained low fouling levels. Unionids are less efficient at differentiating food particles than zebra mussels, and they also have lower filtering rates than zebra mussels. A 2022 study from Texas State University found that unionids suffer energetic stress in the presence of zebra mussels. There was significant glycogen loss in unionids, so this is their energy stores, that were fouled by zebra mussels, but interestingly also in those that were not fouled, but still in the presence of zebra mussels. With respect to the sustained fouling, our data also shows that the highest fouling intensities were recorded during the years of highest unionid mortality. Our data also show that unionid populations began to drop significantly in about 2003, which is 7 years after zebra mussels were found first within the Richelieu River. And that matches up with patterns seen in other invaded waterways, which were those 4 to 8 years of mortality. However, this one did span over a 25-year period to eventually get to those really low levels of unionid populations. It's also possible, of course, that there are other background stressors that work in conjunction with these that we haven't identified and that could be contributing to the mass mortality of unionids that we observed. So given current evidence, we do not believe that any of the alternative hypotheses contributed to the declines of unionids observed in the Richelieu over the past 2 decades. Our results suggest that zebra mussel invasion has caused severe impacts on unionid abundance, even in a river with less than ideal concentrations of dissolved calcium. These results inform risk assessment by showing that waterways with calcium concentrations below 20 milligrams per liter can still support zebra mussel populations enough to cause severe damage to unionids, and also that chronic but low levels of fouling intensities still pose significant risks to unionid populations. Thank you for listening.
Wonderful. Really interesting stuff. Thanks for sharing. I'm just going to share my screen quickly again. Yeah, really great work. Really interesting. I also really enjoyed all of your graphics, but we can get to that at the end. I'm now going to introduce our next presenter. Dr. Krataev is, hold on 1 second. My computer is moving slowly. You all can see my screen, correct?
Correct.
Yep.
Okay. Sorry, it all caught up. Thank you for your patience. So we're going to move on to our next presenter. As a reminder, we will have time at the end for questions for Kennedy, so please use the Q&A to do that. So our next presenter is Dr. Krataev, who is the director of the Great Lakes Center and professor of biology at Buff State University in New York. He received his undergraduate, PhD, and Doctor of Science degrees in aquatic biology from the Belarusian State University. His research interests include ecology, biology, patterns of spread of exotic species and their role in aquatic ecosystems and biodiversity, conservation and management of freshwater ecosystems. He has published over 170 papers and given over 170 presentations at scientific meetings. His research has been funded by numerous federal agencies, including US EPA, US Fish and Wildlife Service, and US National Geographic Society. And with that, I will hand it over to Dr. Krateev. I'll stop sharing my screen and you can pull yours up. Thank you.
Thank you for this introduction. And is it in presenter mode?
Yep, it looks great on my end.
Great. So first of all, I would like to introduce you my courses. Luba Burlakova, Senior Research Scientist in the Great Lakes Center, Vadim Karatayev, Professor in University of Maryland, John Cooper, and Lars Rustam from Cornell Biological Field Station. So mollusks are probably the most charismatic invertebrates in our freshwater. They conduct important ecosystem functions, but also they are most endangered animals worldwide. And in North America, over 3/4 of both unionids and gastropods considered imperiled, and it could be vulnerable, threatened, endangered, or even extinct. They suffered from multiple types of human activity, but habitat destruction and introduction of invasive species, often the most common factors causing deterioration of native communities and extirpation of species. So Oneida Lake is the best studied lake in North America with continuous long-term fishery and ethnology data since 1975. There is a Cornell Biological Field Station on the shore of this lake. It's excellent environment to study and to collaborate among limnologists, and that's why we have a just wonderful long-term data set. Over the years, the lake experienced strong cultural eutrophication, the introduction of invasive species like Dreissenids, round gobies, and few others. Also, the uniqueness of this lake that the first quantitative survey of mollusks in North American freshwater was conducted on Lake Oneida in 1915 and 1917. Then the study was repeated in 60s, in 90s, in 2012 and 2022. Plus, there are, in addition to this very detailed survey, there are annual benchmark surveys on a smaller subset of stations that are going on from 2009 until now. At the beginning of the 20th century, the famous molecologist Frank Collins Baker he spent 2 summer seasons conducting a very detailed study of Oneida Lake, and especially he did very thorough study of in the lower South Bay. What was also great that he published these results of this study in these 2 large volume proceedings, and so we have very good information of what was the molluscan community at the beginning of the 20th century. Very detailed study where each station was located. For example, he wrote that this station was located 50 meters north from the ice house. And we went there and we found the remnants of this ice house. It was very interesting. He also provided photos of many of his samples that we can use to compare what's going on now. So the following surveys were conducted in 67 and 68 by Harman and Forney, in 1995 by Harman, and then in 2012 and 2022-23 by us. So this is a photo of me and Vadim conducted survey in 2012. This is most recent. You can see I gained some gray hair during these 10 years. Luba, of course, didn't change a bit. That's why I have only 1 photo of her. So in both 2012 and 22-23 surveys, we collected samples on 86 stations located in the lower South Bay. And again, this is the best study area that Frank Collins Baker did the beginning of the 20th century, but we also did sample 123 stations in the other part of the lake. Our stations were the same location as in previous studies. We identify all gastropods to species, but spheres only to genus. And we converted all data reported in all studies to common taxonomic resolution using integrated taxonomic index system. So early in the 20th century, lake was in pristine conditions with low phosphorus load. Secchi was pretty good, over 3.5 meters. Macrophytes were very abundant and they spread down to more than 4 meters deep. And so lake was in the clear water phase. And then, of course, like everywhere else, eutrophication started. It peaked in late 60s, early 70s. High phosphorus load, secchi depth dropped to less than 2 meters. Macrophytes shrink in their distribution again, also to less than 2 meters. Lake entered turbid water phase. And then in 91st, zebra mussel invasion, 2004 quagga mussel invasion. They filter water, they improve water clarity. Secchi disk increased more than 3.5 meters, even to the same degree as it used to be at the beginning of the 20th century. Macrophytes became very common and they spread to more than 6 meters depth. Lake again entered the clear water phase. And then in 2013, there was a round goby invasion. So during pristine conditions and the beginning of the 20th century, gastropods were represented by 29 species, almost all native species, only one exotic species, Bithynia tentaculata. Many gastropods family were abundant. See, there's like 7 or 8 species, different families, and each family represented by pretty high density. Also, unionids were abundant and represented by 8 species in the lake. Spheres were diverse, including very large-bodied species of Spherium and Musculum, and this was the only time when they were reported. We never seen them in any of the subsequent surveys. So eutrophication came. Along with the shoreline development and water level fluctuation was strongly associated with a decline in macrophytes. The highly diverse and abundant native gastropod community was reduced dramatically, most likely due to the decline in their primary food source because they're all grazers. They graze on benthic algae and periphyton. And basically what happened that lake-wide gastropod diversity declined by 1967 by 14% and by 1992-95 by 46%. In the lower South Bay, where we have really good quantitative data, the density declined by a factor of 23. So from here to here, and if you look at the gastropod community, you can see that the whole community was represented by one species, Bithynia tentaculata, the exotic one. The density increased 49-fold. This is the only species that can, in addition to grazing, get food by filtering particles from the water column. So eutrophication, lots of food in the water column, the density explode. And of course, Bithynia became the dominant species, represented 95% of all gastropods. If you look at the dominant curve of gastropods in the lower South Bay, you can see that early in the 20th century, it was a very nice dominant curve on lots of other subdominant species. And then in 60s, it became very flat, mostly determined by Bithynia. Other species didn't add much to the dominance. And NMDS ordinated plots show that mollusk community became significantly different compared to the 20s, to the beginning of the 20th century. At the same time, eutrophication didn't cause any changes in the frequency of occurrence of diversity of unionids. And then the Dreissenids came. The introduction of Dreissenids was associated, as already mentioned at the beginning of my talk, with a significant increase in secchi depth, macrophyte coverage. Benthic and periphyte and algae became very abundant, and it increased food base for unionids. In the lower South Bay, gastropod density in 2012 was increased dramatically and wasn't different from the beginning of the 20th century, but significantly different from the time of cultural eutrophication. Again, if we look to the community, many gastropods family became very, very dense, represented by many species in high density. And the dominance curve in 2012 became very similar to 2017. And same was with NMDS analysis that showed that community became similar to the beginning of the 20th century, but significantly different from the peak of cultural eutrophication. So this was the introduction of Dreissenids, was a great message for gastropods. However, the impact of Dreissena polymorpha on unionids was very negative and very quick. Most clams were already dead by 1993, only 2 years after the initial discovery of zebra mussel in the lake. And by 1995, they all were dead. Interestingly, that we never found a single live unionid thereafter. Although unionids are community pretty abundant in the incoming tributaries. For example, you go upstream to a fish creek, maybe half a mile, and there are lots of live unionids. So this is usually the introduction of Dreissenids, especially zebra mussel, caused decline in unionid diversity, density, but very rare it caused complete extirpation that lasts for a relatively long period of time. So, and then round goby came. Their arrival in 2014 had a strong negative impact on virtually all gastropod species during first 2 or 3 years. So very quick. This red line represents round goby increase, and these are unionids, gastropods. You can see that they drop dramatically. And this is true for all, virtually all gastropod family. As a benthic feeder, round goby actively feeds on bottom invertebrates, including various molluscs, gastropods, Dreissenids, and you can see on this movie how they feed on Dreissenids that were crashed when the video system was lowered on the bedrock. So that's why the observed decline in molluscan density was not surprising. What was surprising, however, a partial significant recovery in most gastropods between 3 to 7 years since goby invasion. You can see these red lines indicate significant recovery. Same happened with Dreissenids. The gobies had very strong impact on Dreissenids. Density start decline, and there was not only decline in density, but the effect was to some extent species specific. The higher decline quagga mussel experience. They cannot attach as strong to substrate as zebra mussel. Therefore, gobies and other fish predators prefer feed on quagga than on zebra mussel. And you can see there is some increase in zebra mussel, the relative proportion, but we also can see some recovery about 7 years after goby invasion. The other interesting thing that mollusk recovery was depth related, being limited to deeper areas in almost each of these graphs, they come mollusk recovered and moved to deeper areas because of gobies most abundant on very shallow areas on hot substrate. So the effect of gobies was not only just decline in molluscan density, but also there was an effect on the spatial distribution. And to conclude this part of my talk, just want to mention that over past 100 years, molluscan community of Oneida Lake experienced effects of eutrophication, multiple invasions, including zebra, quagga, and gobies, and overall the impacts of multiple stressors resulted in decline in species diversity and density of native gastropods and extirpation of unionids, spherium, and musculium. Dreissenid mussels currently comprise 89% of the density. At the beginning of the 20th century, 99% of all mollusks were represented by native species, only 1% by exotic. Now we have 89% of exotic species and only 11% of native. So while individual stressors have taxon-specific and sometimes positive impact, like Dreissenid impact on indirect impact on gastropods, the combined effect of eutrophication and species invasion have collectively decimated the native molluscan community of the past century. And this is a photo of what we have now, the typical sample, and this is what was over 100 years ago. And now I want to spend a few more minutes talking about a similar project to some extent when we analyze the effect of multiple invasion, how they can affect the recovery of ecosystem. This was a very large collaborative project, and there are several questions that we asked during this project. How quickly does the zebra mussel impact manifest in each ecosystem feature? Do the impact decline after the initial peak due to complex invasion dynamics or when native community adapt to invader? And in system experience a subsequent invasion by quagga mussel, we want to see whether serial invasions of closely related competing species magnify the impact of the initial invader. So we collected data from 7 different lakes located in Europe and in United States, including Lake Oneida that I was talking just about. And we'll look at what's going on with water transparency, total phosphorus, chlorophyll A concentration, microphytes coverage, and phytoplankton, zooplankton, and benthic biomass. All lakes were initially colonized with zebra, and them were later colonized quagga mussels. So we found a strong ecosystem change that peaked 5 to 10 years of the initial zebra mussel invasion, include significant increase in seki, decline in chlorophyll, decline in phytoplankton, increase in benthos, decline in zooplankton, increase in macrophytes coverage, and of course decline in total phosphorus. These changes characterize a shift from a turbid to a clear water phase and a net transfer of resources from the plankton community to the benthos. There is a special term, a term that called benthification of systems that were colonized by zebra mussel. So following initial invasion, it was interesting to find that there was a partial recovery of plankton community. In about 15 years after the initial invasion, you can see the phytoplankton increase, zooplankton increase, and also it was associated with a decline in seki. And then quagga mussel invaded 4 of these lakes. As you can see on this graph, this is Oneida Lake, quagga mussel form higher density. Lake Balaton, it's only eastern part of the lake, so it's also density became way higher. And in all 4 lakes, quagga mussel increased total Dreissenid biomass and therefore total filtering capacity, and further depleted chlorophyll. Secchi disk went up, chlorophyll decline, phytoplankton decline, zoobenthos decline, zooplankton decline. Unfortunately, we don't have long enough data to see if there was a subsequent recovery of ecosystem. But in conclusion, 7 polymetic shallow lakes with long-term data sets reveal remarkably similar trends, with the strongest ecosystem impacts occurring within 5 to 10 years of zebra mussel invasion. And this is very interesting, and I think it's very important from the management view, because of usually when zebra mussel introduced into the lake, there is a huge changes, and people project these changes for many years to come. But actually, the effect lasts only, the strongest effect lasts only first 5-10 years, and then we can see a partial significant recovery. But the recovery was absent, and impacts of initial invasion amplified in 4 lakes where quagga mussel outcompete zebra mussel and more completely depleted phytoplankton. Right now, quagga mussel spreading across North America and Europe, colonizing lots of lakes that were already initially colonized by zebra mussel and amplifying the effect. For example, in North America, there are about 800 lakes colonized by zebra mussel and less than 100 colonized by quagga mussel, but they're still spreading. And this reemphasizes that prevented invasive species spread is the most reliable means of minimizing their landscape level impact. So there are some knowledge gaps, because what I mentioned, what I was talking about, the impact of zebra and quagga mussel invasion was just polymictic lakes, shallow lakes. What's going on with large stratified lakes like Great Lakes? In these lakes, quagga mussel form much higher density, usually at least by an order of magnitude than zebra mussel, and therefore the ecosystem impact may be stronger. Alternatively, the bulk of quagga mussel population now in all Great Lakes located below the thermocline, isolated from the surface water for most of the growing season, and therefore the impact on planktonic community may be lower than that of zebra mussel. Also, in contrast to polymictic lakes, in stratified lakes, it may take longer for quagga mussel to reach population maximum. Like in Great Lakes, it took them about 25 years to reach maximum. And therefore, the ecosystem impact may be delayed. Recently, we're working on, again, with a large collaborative project. We assembled a large data set of stratified lakes, including Great Lakes, Finger Lakes, European pre-alpine lakes that were recently colonized by quagga mussel, like Lake Constant, Geneva, Lake Zurich, and other lakes, to address questions of the time scale of ecosystem recovery from zebra and quagga mussel invasion in deep stratified lakes. And this is all that I have, and I would like to answer your questions if you have any. Thank you very much.
Wonderful. Wow, what a breadth of information. That was amazing. Thank you so much. So that concludes our 2 presentations for today. We're going to move on to answering some questions questions from the Q&A function. So as a reminder, please put your questions in the Q&A. I'm going to check that. Looks like we're still waiting for some questions to roll in. I did have a question for Sasha. Sure. When you were showing that graph of a natal lake, and you showed that the Dreissenids came in the gobies came in and they decreased Dreissenids. And then there was that blip in recent years where the Dreissenids actually rebounded. Do you have speculation as to why they would rebound like that?
That's a very good question. But as you can see, we have recovery in almost all other ecosystem features after the strongest impact. It's very difficult to say why, because of gobies density didn't decline. They fluctuate widely, but didn't decline. But zebra mussel and quagga, mostly quagga mussel, after the first very strong decline, started recovering, and we're still looking for what's going on there. I don't know. We can speculate that probably because of they somehow adopt to live, and they also their reproduction potential is huge. Usually less than 0.1% of larvae survive to this adult stage. So there is a constant supply of larvae, and when we compare the Dreissenid density in 2012 and 2022, which is just covered the whole this deep and increase, the density was very similar. So they recovered, but they go deeper, and maybe this is why there was a recovery. So I'm talking about average density. I'm not talking about the distribution. Probably they decline in shallow areas, but then went deeper.
Got it. Yeah, I grew up fishing on Oneida Lake, so it was really interesting to see over time how things had changed, even before I was even born. So really interesting stuff. I did have a question for Kennedy. You mentioned that the zebra mussels like to settle on live over dead mussels. I was just curious, do they know why they would prefer that?
So good question. What I've read is that the water flow of the siphons of the unionids create water currents that makes it a better place to settle for zebra mussels because they can kind of feed off of those currents. And also when they're a little bit more upright, like zebra mussels tend to be really good on concrete walls, is that their waste can use gravity to drop away. So it's a little bit more, from what I've read, more efficient filter feeding.
Interesting. I did not know that. It's very interesting. Okay, it looks like we're still waiting for questions to roll in. I had listen, if there's no questions that come in, I do have a list here. So, oh, just kidding. Questions are coming in. We're going to move to some of our attendees' questions then. For Alexander, for Sasha, with recent detection of golden mussels in California, we have been getting a lot of questions on the species. Are you aware of any instances of Dreissenid and Limnoperna overlap, and any thoughts of potential interactions? Very good question.
Yeah, thank you for this question. We wrote a paper years ago in 2007, and we also, Luba and I, we went to Argentina and did the first quantitative survey of Limnoperna. And we speculate what will be the effect of what will be the outcome of competition but to this point, there was not a single place where these both two species coexist. But from what we'll learn from Limnoperna, their distribution is quite similar to zebra mussel. So they live in lakes in the nearshore zone. They require hard substrate. They don't survive in deep profundal zone. So they similar to zebra mussel, but different from quagga mussel. They also can live at a way lower calcium concentration, lower pH, and lower oxygen because they're more tolerant to higher salinity and to low oxygen. So I think they could be more stronger competitors. However, they might require warmer temperature.
Thanks for that. We have another question in the chat. This looks like it's for Sasha. Of the unionid species that were extirpated from Oneida Lake, looks like it's for both, sorry. Oneida Lake and the Richelieu. Are there any similarities in life history strategy that could make them more susceptible to zebra mussel-caused extirpation?
Well, when we look at the we wrote a review years ago when we look at the effect on if there is a species-specific effect of zebra or quagga mussel invasion on unionids in Great Lakes, and we didn't find any.
I could be wrong, correct me if I'm wrong, but I believe that unionids evolved without dominant macrofouling organisms in most cases. So when zebra and quaggas came in and invaded, they didn't have any evolutionary history dealing with biofouling organisms.
No, not in North America, but in Europe, they coexist in lots of areas. For example, in Azov Sea, there are areas when they co-dominate with Dreissena, and this is native range for both, but these are different species.
That's right. That's right.
Okay, we have another question for Kennedy. Is there evidence for diseases being transmitted from zebra quagga mussels to unionid mussels?
So I don't know about any evidence of diseases transmitted. I didn't really focus my research on any specific diseases of unionids. But I guess it would depend on the mode of transmission. If it's some sort of disease that is able to be carried in the water column, then certainly. Otherwise, if it's something that grows on the shells, if they're fouled on the shells, it could be. But I don't want to lead you astray and lie. So I will stick with the safe bet that it's nothing that I've looked at, but a very interesting question.
I may say a couple of words. We did a long-term study of the zebra and quagga mussel parasite diseases, and we found a long list of parasites and the symbionts, but there was no any evidence of transmission from unionids, from zebra mussel to unionids. There are several parasites that could use as definitive host, fish or birds, but nothing about unionids.
Just a side question off that too. This might be more theoretical, but if there's any kind of parasites from fish that Glaucidia could be infected with on gills or fins, that would be interesting if that is a mode of transmission as well. I don't know if that's possible, but just a thought.
Yeah, interesting speculation. Kennedy, I was curious. I know you talked about zebra mussels mostly. Is there also quaggas in the Richelieu?
No, so thank you for bringing that up. I didn't explicitly state that, but in all of the surveys that myself and my lab and my PI have done in the Richelieu, and from other literature as well, there's been no evidence of quaggas yet. Fingers crossed.
Okay. Yeah, I was curious if maybe it was the quaggas having impact on the unionids, and that was something to consider. But yeah, that makes sense. Interesting. Just looking here in the chat, I'm not seeing any more questions coming in. I had one more question for Sasha. You mentioned that the unionids were in the tributaries, but not actually in the lake. Is that because Dreissenids just don't get up into the tributaries, or?
Yeah, there were no, we spent lots of hours looking for unionids, and for several years we found only single zebra mussel attached to one unionid. Usually they're not, streams are not very good environment for zebra mussel. You will never find them unless there is a lake and upstream that act as a source for zebra mussel larvae or quagga mussel larvae. But for all these years, they did not colonize any of this. We checked several streams. We found only one zebra.
Interesting. I thought it might be that, but good to know. Looks like we have a comment for Kennedy, or no, a question. Are the quaggas in Lake Champlain, which I believe is connected, correct, to Richelieu?
Exactly. So the headwaters of the Richelieu or Lake Champlain, so it flows downstream into the Richelieu. I don't believe there are right now. There are certain zebra mussels certainly invaded earlier. Lake Champlain is a higher calcium concentration, so it's a better habitat than the Richelieu for zebras. I would expect that quaggas would arrive in Lake Champlain before the Richelieu, but I'm embarrassed I don't know that off the top of my head. I don't believe there are, though. I'm surprised. I can confirm no quaggas in Lake Champlain. Okay, as of today, I don't believe they are there.
Wonderful.
It is standard, though, as we've talked about, that zebras normally invade first and quaggas tend to come in later on. So we keep monitoring.
Yeah, I didn't know that. That's interesting. Not seeing any more questions in the chat or the Q&A, and I have exhausted my list of questions. So I just want to say, this is your last chance to put questions in the chat, but I also just wanted to say a big thank you to both of you. I really enjoyed all the graphics in your presentation, Kennedy, and Alexander. Those gobies were just ferocious eating those mussels. So very interesting stuff. And thanks for your presentations. I'm just going to share my screen here and close out the webinar. So yeah, thank you all for attending.
Thank you.
And I just want to remind everyone that the webinar will be recorded and posted on the Invasive Mussel Collaborative website. And if you have any additional questions, you can feel free to reach out to us at the Great Lakes Commission. We're happy to connect you with any of our presenters today. And with that, I will close the webinar. Thank you all for attending.
Thank you. Bye.
Thank you.